So my name is Professor Charles Swanton. I'm a senior group leader at the Crick Institute and Professor of Medical Oncology at University College London Hospital and University College London. So the challenge with lung cancer in never smokers is that the direct cause is unclear. There are several associations, for instance, radon, germline genetics, and pollution, to name a few. These are associations. That doesn't prove causation. So our study, we were keen to understand whether air pollution, which has been known for 2 decades to be associated with lung cancer, is associated with lung cancer in never smokers, first of all, and secondly, whether or not it directly causes or promotes lung cancer in never smokers. So firstly, we aim to understand whether the incidence of EGFR mutant lung cancer commonly occurs in never smokers is associated with rising PM2.5 levels across the planet, these 2.5 micron particulates that are emitted from coal combustion, fossil fuel combustion, transport, etc. And what we found is that there is an association in Taiwan, South Korea, and England between rising PM2.5 levels and incidence of EGFR mutant lung cancer. So again, that proves association, but not causation. So the next step was to prove causation. And here we exposed EGFR and KRAS mutant mouse models to rising concentrations of PM2.5s and asked whether these particulates could enhance or promote tumor formation. And in each of the 3 cases, that's what we found in all 3 animal models. So the next step was to ask, well, how is this happening? I think it's helpful to think about 2 distinct models of tumor initiation. The first is the classical model, which is based on DNA mutagenesis. So environmental carcinogens mutate DNA, and that those mutations may occur in a driver gene and the first cancer cell is born. Now that classical model doesn't fit terribly well with emerging evidence, which include the fact that you can have mutations in normal tissue, healthy cells, and work from Alan Balmain and colleagues that shows that 17 out of 20 environmental carcinogens that cause cancer in mice and humans do not induce DNA mutations. So how are they causing cancer? And so we were particularly intrigued by those data because we know that there is no environmental mutagenic signature in lung cancer in never smokers. So any cause of lung cancer in never smokers would have to be doing this independently of inducing DNA mutations. So what we found is when mice are exposed, and actually humans are exposed to pollution, we see this inflammatory network induced, which includes cytokines like interleukin-1 beta. What we then found is that interleukin-1 beta induces the transdifferentiation, we think, of a cell type into an alveolar type 2 cell, which is a progenitor cell with a renewal capacity. Following that, we wondered whether, if that cell harbored an EGFR mutation, whether it would begin to form a tumor, and that's what we find. In other words, you have to have exposure, you have to have a mutation, EGFR mutation or a KRAS mutation, and you have to have exposure to pollution, but neither alone is sufficient. So the question is, where do these mutations come from? It turns out we acquire these mutations with aging in our normal healthy lung. So we find EGFR and KRAS mutations in over half of biopsies that we look at. So these are mutations that are normally recognized in cancers that drive cancer progression that are found in normal cells of the lung. So where does that leave us in the context of our understanding of tumor initiation? Well, it actually fits perfectly in the model proposed by Isaac Berenblum in 1947 of tumor initiation and elaborated over the last decade and a half, 2 decades by Alan Balmain and colleagues at UCSF. And that is the model of tumor promotion. So you have an initiator, which is your mutagen, which can be an environmental carcinogen, but it can also just be aging. Aging is mutagenic, unfortunately. We acquire mutations with age. And the second step, necessary step, is promotion, the promoter. In this case, is air pollution. I think this is one of the first examples of an environmental carcinogen conforming to that model. And if one thinks of Alan Balmain's work showing that 17 out of 20, in other words, the majority of environmental carcinogens do not cause mutations, then perhaps one might conclude that there are many other environmental carcinogens out there that are working through similar inflammatory networks, leading to expansion of nascent clones with mutations in. Well, I mean, none immediately, but I think the first thing to say is that there are two ways of tackling this problem. The first way of tackling this problem is to reduce the number of mutations or eliminate the mutations. That's very hard, and I'm not sure tractable. So we have to look elsewhere. Second obvious answer is reduce the carcinogen, reduce the environmental carcinogen, i.e. lower pollution levels. And that's also a pretty intractable problem, given that economies depend on coal, fossil fuels, and transport, all of which are contributing to the burden of PM2.5s in our atmosphere. So any reductions in PM2.5s will happen slowly over decades, probably, not fast enough to reduce the burden of disease globally. Incidentally, PM2.5s are responsible for between 8 and 9 million deaths per year. So there is an urgent mandate to lower pollution levels, I think, across the planet. It really is the silent killer, not just in cancer, but cardiovascular disease, strokes, and what have you. So the third opportunity, I think, is thinking about how we could intervene to create a sort of a molecular cancer prevention program where you could understand these inflammatory networks from different environmental carcinogens in different tissues and perhaps intervene and dampen the inflammatory networks down. Now the question is, what are those networks? Do they all involve IL-1 beta? I doubt it. I think they'll be different in every tissue, and they'll possibly be different between environmental carcinogens, but only time will tell.